Now the question here is J chain and secretory part is found in which type of antibody? The options are immunoglobulin D. Okay, and one more important thing which you have to be focused on, that whenever any figure is given, you have to read the details in very carefully. So if you are having presence of mind there, so you don't need to remember all these structures of the antibodies, but you can get the keyword here, that is IgD, IgE and IgA. So the names are written, and same happens with the instruments also. Sometimes the name of the instruments are written on the instrument itself, but we do not pay a heed to that, right? So that has to be very carefully analyzed whatever picture is given. So if we just talk about the keywords here, so we have the J chain and the secretory part. So if we just take the J chain is generally found in two types of immunoglobulin. Immunoglobulin A, which is generally bimeric, or it can be tetrameric as well. Then we have immunoglobulin M, which is a pentameric polymer, and that also has J chain, which joins its various bivalent monomers. Okay, now here another part, that is the secretory part. Now this secretory part is found along with the J chain, is found in your immunoglobulin A. Basically there are two types of the immunoglobulin A. One is your serum immunoglobulin A, serum IgA, and another is your secretory IgA. Now what happens in the secretory IgA, you will be finding a J chain. Okay, it is a glycoprotein basically that will be joining the two monomers of the immunoglobulin A, and it also carries a very small polypeptide thing that is known as your secretory part. Okay, there will be a secretory polypeptide as well that will be helping in the secretion. Now certain important things about the immunoglobulin A itself, which are generally asked in the exam. The first thing that is asked, that is it is the second most abundant antibody found in the body. Okay, then the second important thing which we have to understand, it is most predominantly found in the saliva. Okay, this immunoglobulin A is also found in the milk. It is also secreted in the milk along with immunoglobulin G. Okay, so these are certain important things which are asked in the exam. Then two other important things, very factual things. One is the immunoglobulin A is used in the caries vaccine. And another very important thing is it is used for activation. It is responsible for activation of alternate pathway, alternate complement pathway actually. Okay, so these are various things which we have to remember about the immunoglobulin A. Now one more important thing, that the immunoglobulin A, which is a secretory type, for example, the secretory type, it is generally secreted by the mucosa or glandular epithelium. It is secreted by mucosa or glandular epithelium. It is very important, and this basically protects the immunoglobulin A from proteolysis. So that is very important to understand why it is secreted from mucosa and glandular epithelium, to protect the secretory part or the immunoglobulin A from the proteolysis. So these are very important things about the immunoglobulin A which we have to remember. Now if we just see the picture here, the structure of the secretory immunoglobulin A is given here. So you will be able to appreciate this is one monomer and it is monomer 2, and both are joined by a small glycopeptide chain that is termed as the J chain. And this particular immunoglobulin, which is having a secretory component as well, this one is the secretory component, so it is known as the secretory immunoglobulin A. Okay, now this particular immunoglobulin is synthesized by plasma cells. Okay, now these where are these plasma cells situated? These are situated in the mucosa or the glandular epithelium. So this dimeric epithelium, so this dimeric immunoglobulin A is generally found in the is generally formed by the plasma cells, and it is generally dimeric. Okay, now the J chain, which is used for joining these monomers, it is also formed in the same cells. So that is very important. Sometimes they may ask where is J chain of the IgA is formed. that is also formed in the plasma cells, which is seen in the mucosa or glandular epithelium. Okay. Now, another very important thing about the IgA, that is, we have to just know the schematic diagram of this particular thing. So here you can see this is J chain, this is your Y-shaped immunoglobulin A. Here this one is your heavy chain and the smaller one is your light chain. These heavy chains are joined with each other with the help of two disulfide bonds. Now these two, this is one monomer and monomer number 2, these are joined with each other with the help of a small glycopeptide chain which is 15 kDa thick. Okay, so this is 15 kDa thick, and means this is the weight of the J chain glycopeptide here, and that will be joining the both the components or both the single monomers of the IgA. Here you will be able to appreciate the secretory component which is also joined to these two monomers here. Now, one important thing I must say here, if we just see, because J chain is found in IgA as well as IgM. So if it is found in IgM, it is going to join the 5 monomers of the IgM. Okay, so since it is 15 kDa thick and there will be 5 monomers, that's why it will be a pentamer, right? So each monomer is 160 kDa thick. Okay, and that is going to form a pentamer that would be around 950 kDa thick. So these are certain values which are generally asked in the exam. So if we just go back to the question here, so the J chain as well as the secretory part are found in IgA, that is your option number 3.